cover information in chapter 6 and 7. We will talk about how to grow microorganisms in laboratory conditions, and towards the end of this lecture, we will discuss how to control them. Set it up so that way their growth is successful and those cells do not die. So our bacterial cells, which we'll focus on for most of our microbiology class, one of our bacterial cells, if we have one cell, that cell will eventually divide, and it will divide into 2 cells. All right, so one cell will become 2 new cells. All right, they will double. Right, and what will happen? Those cells will divide, and each cell will double. Right, and what will happen? Those cells will divide again, and those cells will double. So every time the cell divides and gives rise to more cells, we call this process binary fission. Right? So binary fission, this is the reproductive process of bacterial cells. Right? And when one cell divides, right, to make 2 new cells. All right, 2 new identical cells. All right, so the cells, every time they divide, the cells will be identical, will be identical. So since they are identical, that means they all have the same genetics. Same genetic information. All right, so they all have the same genetic information. So when we bring the cells again from an outside or another environment, we want to know how to grow them and how that's done in the laboratory. For our teaching laboratories, we are constantly working with samples like E. coli and Staphylococcus, and we want to make sure that we have enough of them to supply students with the cells that they need to perform their experiments on those bacterial cells. So let's look at the first thing that we need to understand about our bacterial cells. Each bacterium will go through a phase of growth. So once we transfer a bacteria to an environment that is suitable for their growth, then they will grow successfully and they'll continue to divide and double and double and double until you have many, many cells. So let's look at the 4 phases of growth that the bacterial cells go through. The first phase is called the lag phase. All right, and in the lag phase, in this period, this is when we transfer bacteria to their new environment. Are very metabolically active, so they're trying to get as much nutrients from the environment. So at this time, they're trying to get food, right? And they're taking that food and most of it, converting it to building blocks and energy, ATP, to support their growth, right? So the food that they take in, metabolically active, right? They're taking in food, they're breaking it down, and then they will get the building blocks and energy to grow. So this is very important when they get into the new environment, that they check it out, they see what's there, and make sure that what they need is there so that way they can grow. So this varies in the clinical setting. So let's talk about in the clinical setting what this looks like, and then what this looks like for your microbiology lab. We'll put student microbiology lab, right? So we can distinguish how and what is being grown. So in the clinical setting, in this case, if you get a sample from a patient, maybe there is a sick patient, right? And that sick patient will give a urine sample, right? If they have a UTI, right? And that urine sample will go to a clinical lab where they will grow whatever's in the urine. They want to grow it in food. They want to grow it with food. So the food will either be on a petri dish, containing food, or it will be in a test tube. All right? So the test tube will have nutrients, the petri dish will have some sort of nutrients, so that way, all right? So you'll grow it either on petri dish or test tube. But in the microbiology lab, we will have small containers that usually contain our bacterial samples. So maybe it might be in a frozen, the bacteria may be kept as a frozen stock, or maybe, or let's say freeze dried stock. So you might have some sort of stock or preparation of whatever bacterial sample, whether it's E. coli, Bacillus, Subtilis, Staphylococcus, whatever organism you want. And then what will happen, your lab technician will then grow it either on the petri dish, or, so we'll put student, we'll put lab technician, or no, so we'll just put grow, right? So they'll grow it either on petri dish or test tube. So pretty much it's the same thing, except we're not getting random samples from different patients in this case, but we're taking our samples from a supply that we have, so that way we can study these organisms in your lab time. But either way, what we're doing is that we're taking the samples from a condition where maybe they're not growing so well, or there's probably not enough numbers to study, to do stains, simple stains, cram stains, other laboratory tests to identify the organism. We're transferring it to an area of growth, so that way we can have more cells. So once we grow them either on the petri dish or test tube that has food, all right, so again, at this stage, this will be more food, all right, so more growth. And one thing that you want to, when you're giving food would include things like carbohydrates, proteins, lipids, whatever the bacterial needs to grow. And then on top of giving, in addition to giving them food, of course you have to make sure that the other environmental conditions, such as the temperature and pH, will be suitable for the growth of that microorganism. But this gives you an idea of what is going on routine in the clinical setting versus a student lab, where we're getting the samples from, all right? So, and most of the organisms that we work with in our laboratory, we know they're pretty safe for the students. So they're going to be grown on a petri dish or test tube, and then from there, you can take this and you can perform your tests. So you would test the organisms once they have grown. You can then do things like simple stain, or a gram stain, or whatever test that you would like to do. All right, so that gives you an idea of what we're doing. But most important is that the organism has to grow, right? They have to grow in this particular setting, and they have to divide. Once they're on the petri dish or in the test tube, they have to divide. And that's providing, that's of course considering that the environmental conditions are going to be good for them, right? So again, they want food. So not just food, they're looking to see that so one thing they're looking for maybe is food. Of course, they want to make sure that the temperature is good for them to grow, right? It's not going to destabilize their cell. And the other thing would be maybe the pH, also the pH, right, for that environment is suitable for their growth. So there's some things that are important that they're checking out at that time. Okay, so the next stage of growth, which is going to be our log phase, this is when binary fission occurs. So as I mentioned, with binary fission, one cell, right, will divide into two new cells, and those two cells will divide, and they will double, right? Each cell doubles, and they will divide again, and they will continue to double as long as the environment is suitable for their growth. Right? There's enough nutrients, there's enough pH, there's enough temperature, there's no toxic materials in their environment. So when they double, every time they double, we call this a generation. Right? And when these two cells double to 4 cells, this will be another generation. And when these 4 cells double to 8 cells, this will be another generation. Okay? So, the generation that we start out with for this one cell, right, this one bacterium, this will be our first generation. And when those cells double, that will be the second generation. And when those cells double, that will be the third generation, and so on. And they will continue to divide as long as the environmental conditions are stable and suitable for their growth. Is there enough food? Is the temperature right? Is the pH right for their growth? Different cells divide at different rates. So, for example, E. coli divides, and I'm going to underline for scientific nomenclature, this organism will divide or double about every 25 minutes. All right, approximately. So I'll just put like that, right? Approximately every 25 minutes. And something like Staphylococcus aureus, and I'll underline the scientific nomenclature, divides or doubles every 20 minutes. Okay. So if we're talking about Staphylococcus, their first generation or generation time will be 20 minutes. Right? For S. aureus. And how much time will it take them to complete their third generation? Yes, so it will be about 60 minutes. Right, so we multiplied, we went through 3 generations times 20, that would be a total of 60 minutes. That means in order to complete your second generation, how much would it be? Yes, so it will be 40 minutes. And of course, for E. coli, it will be different, right? Their first generation will be 25 minutes, so how long will it take them to complete their third generation? Right, it will be instead 75 minutes, all right? So the growth time of different bacteria, the time it takes them to divide, will be different for each cell. All right, so we can continue drawing out a map like this to figure out how many cells that we'll have after, let's say, 24 hours or 12 hours, but we have a formula. So we use this formula here, the number of cells equals x, multiply x times 2, to the n, and n is an exponent. So there's several things that we need to consider when we're using this formula. One thing, how many cells do we start out with? Do we start out with 1 bacterium, or do we start out with 3 or 5 bacteria? Because that's going to affect the number of bacteria that you put with food, right? It's going to affect how many you get at the end. If you put 1, right, you're going to have less bacteria after 12 hours, but if you put 5 cells, right, you of course after 12 hours you'll have more. And then we want to be able to calculate the number of generations. And the number of generations, you have to figure that out. Sometimes it may tell you, or you have to figure it out based on the time that it takes for each cell to divide. Right? So let's look at one question involving our growth, all right? So the growth of the cell. So it says in our question, 4 staphylococcal cells are placed in nutrients. So you're giving them food, solution, some sort of broth. So likely this is in a test tube, and each cell divides every 20 minutes. We're trying to figure out how many cells are present after 100 minutes. So when we start out, 1 cell divides into 2 cells, so that will give us our first generation. And our first generation, of course, since it says that each staphylococcal cell divides every 20 minutes, we know that this first generation took 20 minutes, right? And when they double again, right, this second generation, of course, will take another 20 minutes, so that will be 40 minutes, right, that we have here. And of course, so it will be 40 minutes, but in this question, we're starting out with 4 cells. So of course, that would mean that we will get, if we have 4 cells, we will get more bacteria at the end of growth, as opposed to starting out with 1 cell, right? So we want to know a total of how many generations, because we're beyond 40 minutes, right? They want to know how many do we have after 100 minutes, and how many cells will we have, right? So how many staphylococcal cells are present after 100 minutes. All right, so this is the formula that we use. We took this from the previous page just to figure out how many cells do we have. All right, so you need to figure out what is X and what is N, all right, for your formula. So what is X? How do you figure out X? You read the question. It tells you that you're starting out with 4 staphylococcal cells, right? So this number will be 4. And how many generations, right? We know that we're beyond 2 generations because they said 100 minutes. So in order to calculate the number of generations, we have to look at the generation time. And we would divide, so for N, you would divide 100 divided by 20, right? To figure out that there are 5 generations. Right? So in total, there are going to be 5 20-minute periods in 100 minutes, right? So 5 20-minute periods. So if we add on another 20 minutes here, right, we'll get 60 minutes for the 3rd generation. Right, for the 4th generation, how many minutes will we be at? 80 minutes. And for the 5th generation, now we're at 100 minutes. All right, so in order, again, taking the total amount of time and dividing it by how many minutes it takes them to double, and that would be 5 generations. So the answer here, I'll just erase this, just so for 5, so we know N is 5. So we plug everything in, right? So we do X as far as order of operations. You would execute your exponents first, and you want to practice this at home. Sometimes on exam, because it's so quick, right? You don't want to make any mistakes, so you have to practice at home. So I would multiply. You can't use calculators on your exam, so you need to practice what 2 to the exponent 5 means. So what does that mean? We're multiplying multiples of 2 5 times. So it's 2 times 2 times 2 times 2 times 2. So 1, 2, 3, 4, 5. Multiples of 2's 5 times. So we would multiply the exponents first. So we're multiplying 2 times 2, which will give us 4. All right, and I crossed it out. Then I'm multiplying 2 times 4, and that gives me 8. So I cross out that 2. Then it's 2 times 8. This will give me 16. I use that 2, I cross it out. And then 2 times 16, right, gives me 32. So I cross out that 2. So now I know I multiplied all my 2's. I didn't leave any behind. So my exponent 2 to the 5th will give me 32. And now I multiply it times 4. All right, and when I multiply that times 4, that will give me 128 cells. All right, so 4, I'll just bring this down, 4 times 32. Right, and then, all right, it's good to kind of circle and label everything so you know what exactly you've done. All right, and on the exam, you would be able to answer a question like this. All right, so let's try one more question similar so that way you get a little bit more practice. And this question was from Pearson, Mastering Microbiology. All right, so here we have this question. All right, pretty similar. Maybe some of you guys guessed on mastering, didn't know why you got the correct answer, but many of you guys did not ask any questions to figure out how to do it, right? So you need to know what you're doing and why you're doing it, especially in this class, so that way you can answer questions and get them correct, right? It will give you advantage if you understand why or understand what it is you're doing, not just guessing, oh, I got it right, and you don't know why. So let's try this question. I'll give everyone a moment to look at this one and figure it out. Okay, so for this question, what do we need to do? The first thing, you need to know your formula. So start out with the formula, number of cells equals x, right? 2 to the n, right? N is the exponent. You need to know what x and n is, right? X equals, and every time you do it at this point, you should write everything out so that way you will remember this by the time you get to the exam. So x equals, yes, so x equals the number of cells. And what is n? So numbers, yes, you start with. And n would be what? So n would be the number of generations in total that this cell has gone through after division. So what do they tell you in this? So it says 3 cells with the generation time of 68 minutes. So from the question, you start out with 3 cells. So x equals 6, all right? So you have that. So they are inoculated. Inoculated means transferred, all right? So inoculated means transferred. I'll write that term here. And they are transferred, so they are transferred to a culture medium. All right, so culture medium would be the nutrients that will be provided to them. All right, so transferred, sorry, spelling here, culture medium. We're talking about nutrients, right, or the food. All right, so we're trying to figure out how many cells are present after 5 hours. So they tell you how long it takes for one cell to double, all right, the generate or divide, right? So that takes 60 minutes, and we're trying to figure out how many are there present after 5 hours. So for the number of generations, they're telling you that one cell divides, so one cell, so we know that our one generation, right? Generation 1 will take, generation 1 will take 60 minutes. minutes will be in 5 hours, right? So in order to do this, you multiply 60 times 5, so you'll have a total of 300 minutes, right? But the question is looking to find out how many generations. So we're still trying to find out how many generations. So we're trying to find out how many generations have occurred after 300 minutes. All right, so now it becomes similar to the question before. We will divide 300 by what? By 60, right? And that gives us 5 generations, all right? So we have a total of 5 generations. So now we have our number for X, and we have our number for N, all right? So we can go ahead and plug in the numbers for our formula. So it's X is 6 times 2 to the N, which is 5, right? So it will look like this. So we can practice our multiplication here again. So we're going to make sure we do our exponents first. Remember, multiples of 2, 5 times. So 1, 2, 3. Do I have enough 2's? No, I need 2 more. And practice the multiplication when you're doing this. So 2 times 2 would give you 4, cross it out, right? 2 times 4 will give you 8, right? Cross that out. 2 times 8 will give you 16, cross that out, right? 2 times 16, 32, again, cross it out. Now we know we use all of our 2's, especially on an exam. We want to make sure that we are definitely sure, and we multiply that times 6, and that will give us a total of 96 cells after 5 hours. And the time, and we want to grow this for, let's say, about a day. So here would be at time 0, right, or time 1 hour, right? And this will be about 24 hours, all right? So at time, when we start out with, it's somewhere, let's say the number of cells, maybe somewhere, let's see, let's map this here, number of cells. Maybe it's somewhere, so let's see, hold on. Answer is, we said 96, right? So after 5 hours. So if we were to plot how many cells divide over time, relatively, if we start out around an hour or so, we know the growth will be somewhere around here. Now I'm just going to use my calculator to quickly get through or look through I'm going to use a calculator on my phone to quickly plot how many cells we'll have after 24 hours. So of course, if we're doing for 6 hours, right, it will be 2. For 6 hours, it will be 2 to the 6, right? The 6 generations times 3 for 6 hours. And for 12 hours, it will be 2 to the 12 times 3. So this will be for 6 hours. I'm going to do for 12 hours, just some even time points here. And then for 18 hours, right? 18 generations, right here. So that's for 18 hours. Right, and then for 24 hours, right, it will be the exponent of 24, because it's how many generations, right, after that time, 2 to the 24, all right? So just to calculate the number, so I'm just going to use my calculator on my phone to get this, and I'm going to use, in order to calculate the exponent, I'm going to use the symbol on my, I'll write it here, x to the y, so this is the function that I'm putting in. So let me just put a box around it, so that way we know that that's what I'm putting in on the calculator. So what I would do is I'll put 2, and then I'll press the symbol on my calculator to the 6, and that will give me 64, and then I'll multiply times 3. So at the end of 6 hours, I'll have 192 cells. Then I'm going to do for 12 hours, so 2. I'm going to use that symbol on the calculator, and then press 12, right? Because that's the number of generations. That will give me 4,096, and I'm going to multiply times 3 because we started out with 3 cells. So that will give me 12,288, right? And that's how many cells that I will have there. And then after 18 hours, or 18 generations, I'll do the same thing. So 2, I'm pressing that symbol on the calculator, then I'm putting in 18. That gives me 262,144, and I'm going to multiply that times 3. 18 times 3, so that should give you about 786,432 cells. And then after 24 hours, I'm going to do the same thing, 2, and then I'm going to press that symbol on my calculator, put 24 in. That's going to give me 16,777,216. So I'm going to have that and then multiply times 3 because I started with 3 cells. So it will look something like this. So over 50 million cells after that time. So if I were to plot that, right, remember we started out with 3 cells, so it's all the way very close to not that many, right? So because this timeline is in the measure of millions of cells, and then at about 6 hours, I have 192, still very small. I'm going to put the dot somewhere around here, right? Because you could barely, right? This is representing 25 million cells. Around 18 hours again. Wait, did we do that? No, sorry. Around 6 hours, 192. So that's still very low, right? At that time point, sorry, for 12 hours, again, still very low. And then for 18 hours, again, still very negligible here. So I'm just going to put this a little bit so you can see maybe a slight difference. But then for 24 hours, what happens? We're all the way up here with millions of cells that we can work with if we were, like I said, to perform experiments in a gram spinning lab. So if we were to plot this exponential curve, what happens? We would see a trend like this, all right, which is hard to understand. So this is our exponential curve, right? It's hard to understand the trend as far as how the bacterial cells are growing, right? Right? Because at the end of 24 hours, it looks like all of a sudden it just shoots up, all right? And we really can't understand that. So in science, especially when we're plotting the number of cells here, we often use what is called a log curve, all right? So log would be the log phase, all right? So if we're looking at the log, right, usually we'll have time, and I'm just going to put my time here, hours, right? And for the number of, so we'll have 0, 6, let's see, 18, 12, and 24. So it should look something like that. All right, so so if we're looking at this, we're going to plot the log. And the log, when we plot a log curve, the log curve gives us a better, a semi-log curve, it gives us a better idea of how the cells are growing. So we're going to put here logarithmic log number of cells. And we're going to do this on a scale of about 5, let's say till 10, right? So something like that will give us will give us an idea of what this will look like, right? So for this in the calculator, what you can do is use, so I'm going to look at my calculator just to get the function that we'll use, and you could use log 10. So it will look something like this. All right, right? So this is for the exponent, when we're transferring exponents to the semi-log curve, we use that, all right? So I'm going to put in my numbers here. So my first number, I'm going to convert 192, right, to my semi-log number. To my semi-log curve. So 192, and then I'm going to put that in. So it's going to give me about 2. All right, so here we are about 6. All right, so let's say somewhere about here, 2.28. And then for 18, make this right, oh, sorry, I was backwards, so 12, 18. So for 12 hours, I'm going to take this number here, 12288, and I'm going to press that function, which is here, all right, on my calculator, and it's going to give me about 4. So 4 will be somewhere roughly around there. All right, and then for 8, I'm going to do the same thing for the number of cells that I got for 18, 786, 432. Again, I'm going to press this button on the calculator, and that's going to give me about 5.98, so I'm somewhere up here, maybe. And then for 24, right, I'm going to use the number of cells here, right, to get my logarithmic number of cells. So I'm going to put in 5331, 648, and I'm going to press my symbol here on the calculator, and that's going to give me around, somewhere around like 7-ish. So we're probably about 7.78, so we're somewhere about here. And once you have your numbers in the log, what that allows you to do is draw a curve, a straight line, and this log, when we're looking at the line of our semi-log graph, it gives us a better trend that the cells are going to be increasing at a steady rate, sort of at a steady rate. This is harder, the exponential curve is hard to see that the cells are growing, especially down here where the numbers are so close, where the plot is so close together. So again, for demonstrating the growth of cells, we use in science and especially in biology, we will convert our exponential curve to our semi-log curve, and this allows us to see how fast the bacterial cells are growing if we convert it to the log curve. Here again, it's hard to see that there's an increase in the number of cells, but we can look at this curve and see there that there's an increase. So we use this representation, the log number of cells, to look at the trend. So all this is, when we're talking about the log phase, they're taking the exponential numbers and putting it into a linear log curve, so that way you can see the increased number of cells. So just going back a few slides to where we were here at the log phase, right? So remember, the lag phase is when the cells are just checking out if the surroundings are suitable for their growth, and in the log phase, this is where they're dividing, and this is where microorganisms are most sensitive to anything that you might add in their external environment. If you add any antibiotics, inhibitors, any drugs that can harm their growth, this is the time that it's most effective. So if you're taking an antibiotic, it usually will target the cell as they're dividing, and it will block them from making structures for the new cell, as far as maybe the peptidoglycan cell wall, making the plasma membrane, making ribosomes. This is when it will target those structures as they're building them back up for each new cell that they're making every time they double. Okay. The third phase of growth is the stationary phase, and this is when the cells start to encounter stress. And stress comes comes when there's too many cells, and all those cells are depleting the amount of food that is available. All right, and with having so many cells there, the oxygen levels are now taken up, and if there's any water in your nutrients, that is being used up. So the cells are using everything, all those growing cells are using everything up. And of course, as they're taking in stuff, taking in this food, they are going to be releasing lots of waste, right? And that waste is also going to cause the pH of that environment to change. So the cells don't like it, and they will not be able to survive. Some cells have ability to get through the stationary phase and live on to get through another cycle of growth. And these cells may tend to form endospores. There are two species of organisms that were discussed in lecture 2 that can form endospores. Do you remember what they are? So yes, one will be bacillus. So we have bacillus species. I'm going to write bacillus, capital B-L-L-U-S. Bacillus, underline it. SP stands for species, so that's not underlined. Right? We have different bacillus species. We have Bacillus subtilis. We have Bacillus anthracis. All right, so there's different organisms that exist, and we have one more group of organisms that, one more species of organisms that can form endospores that we'll take a look at in our class. It is mentioned in lecture 2. It begins with a C, so that is Clostridium. All right, so the Clostridium species. All right, so that is our organism that we have. All right, other organisms do not form endospores, like E. coli, Staphylococcus. They don't form endospore, but they may be able to store things that may help them to survive a little bit longer, or possibly go through another cycle of growth. For those cells that do not go through any more growth, they will enter into the death phase. All right, so this is when they die, and of course, the cells do not have the conditions that are needed for them to grow any longer. So cells like E. coli and Staphylococcus that we started out talking about, those cells, again, after a couple hours, you'll have maybe a few cells, 100 cells or so forth. But again, remember, that's not enough to work with. Usually when we grow E. coli and Staphylococcus in the lab, we grow them overnight. And by the time they grow overnight, more or less for 12 or close to maybe between 12 and 24 hours, right, we'll have millions of cells, and by that time, we would know we have many, many cells to work with. But if the cells continue to grow and we let them grow beyond 24 hours, what will happen? Those cells will start to die because there's way too many cells. They'll encounter stress, and the changes in the environment will be too much for them to deal with, and they will not be able to survive that condition. They will not be able to survive those conditions. So when we're, again, one of the factors that we need to think about when we're growing our cells, we need to know how long does it take them to double. And we can find this information, it's for most cells that we work with in our microbiology laboratory, most of it is published. But if you don't know how long it takes for them to double, you would have to run experiments to figure that out. So the next thing you want to consider when these organisms are growing, you would want to consider, do they need oxygen or not? And in your homework exercises, they give you this setup of these test tubes, sort of explaining the growth of these different groups of organisms with or without oxygen. So the way how the tubes are represented, each tube will correspond to different things. So the top of the tube means that is relating or trying to tell you that they're representing where the oxygen levels are the greatest. And the bottom of the tube where there's no oxygen or very low oxygen. So our two contrasting organisms, our obligate aerobes and obligate anaerobes, are here. We have our obligate aerobes, which they show you in this tube on your left here, right, it has organisms and they're all the way towards the top of the tube, again representing where oxygen is the highest. But when we compare that to our obligate anaerobes, right, they're completely opposite. They don't want any oxygen, so they're at that end of the tube. All right, so what's the difference between these organisms? Why can one tolerate oxygen and one cannot? Well, these organisms, if we look at it, the obligate anaerobes, what happens with them is that when oxygen is in the environment, oxygen gas, which is, so oxygen gas that is in the environment, O2, 2 is a subscript, so this is the gas that is natural or normal, I should say normal in the environment, but what happens, oxygen becomes, I'll just put oxygen abnormal. Okay, so oxygen has the ability to become into this abnormal state. And when it becomes abnormal, what happens is that it will hurt the cells. It can harm cell structures. Right? So it can harm cell structures. So these obligate anaerobes cannot deal with this because the cell structures are damaged, and they do not survive. But the obligate aerobes, what happens, they have special enzymes, superoxide dismutase and catalase in their cells, and those enzymes allow them to take these toxic forms of oxygen and make them good, and make them or convert them back into the normal form. Right, so they'll take the abnormal form of oxygen, and now the obligate aerobes are able to convert it back this way. All right, so the enzymes allow it to go reverse the state of oxygen back to its normal state. So you need the enzymes in order to do this. And only the obligate aerobes have those enzymes, so they're able to take the abnormal oxygen and make it normal again, so that way it's not hurting their cells any longer. Okay, so again, when we're growing these organisms, we need to be careful that we're either giving them oxygen or not giving them oxygen if they need it, or they do not need it when they're growing. Okay, and when we talk about this later on in metabolism, about growth, the requirement for oxygen, we'll talk about that a little bit more in detail. Okay, so your facultative anaerobes are present here, and the facultative anaerobes include many of our familiar organisms in the lab, and these organisms can use free oxygen, but they also can grow without it. And your aerotolerant anaerobes, similarly, they never use free oxygen and are indifferent to it. And if you look at their tubes at the bottom, you can look at their growth and see if you can sort of identify or look at the difference between the way how they're growing in the tube. So if you notice with your facultative anaerobes, these organisms, again, they can grow with or without oxygen. And if you notice, many of them, they want to grow, or them going or present where oxygen is higher on the tube, as you see in this representation. But with your aerotolerant anaerobes, when we look at them, they can grow anywhere. and it doesn't matter. They're indifferent to oxygen, and they are not affected by oxygen. So again, the facultative organisms, if oxygen is available, they'll use it, and they'll grow better in the presence of oxygen. However, when we're talking about the aerotolerant, they're not using it to grow, and they can grow whether it's there or not. Indifferent means it doesn't matter if it's present. And then we have our microaerophiles like spirillum, a small spiral-shaped organism. This organism requires small amounts of quantities of oxygen,